In today's video lecture, I'm going to talk about lupus nephritis. And as you know that lupus nephritis is a type of kidney disease caused by systemic lupus erythematosus, which is an autoimmune disorder, and the kidney disease caused by lupus may get actually worse over time and lead to kidney failure. And lupus nephritis occurs in 5 out of 10 adults with systemic lupus erythematosus, and in case of children with SLE, actually 8 out of 10 will have kidney disease. And as you know that lupus nephritis is an autoimmune disorder, so the predisposing factors to all autoimmune disorders, it is not a single factor. Actually, it is a constellation of factors, as I mentioned in a previous lecture. Genetic predisposition, it is not enough to get the disease, but however the genetic predisposition, it needs an environmental factor, for example infection. And as it has been shown that several genes have been associated with lupus nephritis, for example HLA, DR3 and DR15. As can be shown here, lupus nephritis occurs when the expression associated genes increased in some individuals. Actually, these associated genes, when the expression get increased, a patient may get lupus nephritis. However, nucleic acid and DNA, double-stranded DNA or remnants of the nuclei, when they are circulating within the circulatory system due to failure of apoptosis. Actually, in case of apoptosis, all remnant of apoptotic products are removed by a special subset of macrophages. But however, due to failure of apoptosis, the remnants of nuclei and the gene products and nucleic acid exposure may lead to formation of specific antibodies. Also, as can be seen here, lots of antibodies here, and this is the glomerulus, and this is the basement membrane, and here this is the glomerular capillary, and as can be seen here, there are large amount of antibodies that are produced in situ, or they come through the circulation. However, these pathogenic antibodies together with formation of immune complexes. These are the immune complexes, as can be seen here. All these are immune complex. The immune complex means antibody plus antigen or auto antigen. So these are the key links leading to lupus nephritis. The source of these immune complexes may be through the circulation. They arrive to the kidney and to the glomerulus or the glomeruli, but however, immune complexes can be produced in situ, and they bind to the endothelial cells of the glomerular capillary, and this will lead to activation of the complement with subsequent accelerating cell damage. But however, as can be seen here, these are the podocytes. Here they are appear. This is the basement membrane. These are the endothelial cells of the glomerular capillary, and these are the podocytes. So the position of immune complexes below the endothelial cells or as the endothelial area, this will lead to proliferation of cell here, or a proliferative disease and glomerular crescent formation, and accordingly podocyte get according to the proliferation of cells here, this podocyte get distracted, and this may lead to variable or varying degrees of proteinuria. So now the area, it is an inflammatory area due to migration of immune complexes to the kidney, or due to formation of immune complexes in situ. So this inflammatory area leads to recruitment of more cells. For example, we have B cell, T cell, dendritic cells, and also we have CD8 and T helper 17 T cells. And as can be seen here, that with regard to neutrophils, which may be the very important player in pathogenesis of lupus nephritis. So during lupus nephritis, levels of neutrophils get elevated, and neutrophils in SLE patient get activated, as can be seen here, intravascularly, and overexpress various adhesion molecules, and display tendency to aggregate, I mean the neutrophils, and also neutrophils in SLE are characterized by increased netosis. As can be seen here, the neutrophils, they modify their chromatin, and they send some threads or sticky projections. During normal infection, these are very important for trapping the microorganism. So these now are from neutrophil, and they are extracellular, as can be seen here, traps. It looks like they trap. But however, in case of lupus nephritis, the situation may get worse with this phenomenon, as netosis or formation of these nets or neutrophil traps, or extra neutrophil, extracellular traps may play an important role in pathogenesis of SLE and may deteriorate the situation. And however, this netosis, as I mentioned, may induce formation of interferon alpha. We have these cytokines, interferon alpha, we have TNF alpha, and activate immunoinflammatory cells and activates the blood coagulation system and contribute to formation of blood clots, causing cause endothelial dysfunction and tissue damage. This is all due to netosis, or formation of neutrophil extracellular traps during the inflammatory process as a result of formation of immune complex. And also, as can be seen here, this is reactive oxygen species. So this reactive oxygen species, actually the source are the cells and vascular endothelium. The source of this is the vascular endothelium, and these cells are the source of reactive oxygen species, and this lead to activation of T cells. So here we have T cell, we have T helper cell, we have CD8 cell, we have T helper 17, and these are actually, they have chemotactic. So these are chemokines produce or can chemo attract other cells to the area, and this will lead to more deterioration in the condition, and this is due to formation and production of reactive oxygen species. And as can be seen here, this T cell, we have here T cell, B cell, dendritic cell, and plasma cell. All these contribute to polyclonal immunity, and the intrarenal etiology of lupus nephritis involves also antibodies, as can be seen here. These antibodies within the glomerulus or the capillary that bind to glomerular capillary to the endothelial cells, and involves antibody binding to multiple intrarenal autoantigen rather than deposition of circulating immune complexes. And also CD8 and T helper 17 T cell as well as B cell infiltrate the kidney in lupus nephritis. Recently, some advances have been made for developing an effective treatment, and so far 2 effective medications, they are approved by FDA in treating nephritis, and this include Benlysta. These are monoclonal antibodies, and a monoclonal antibody targeting B cell survival, down-regulates the activity of B cell, and as you know that B cell is a source of autoantibodies. So by down-regulation the activity of B cell, this will down-regulate the production of autoantibodies, and so if the load of antibodies are low, so no much immune complexes will will form and the patient may improve. Also other medication known as voclosporin, and it is an analog to cyclosporin, has been approved by the FDA for treatment of SLE and lupus nephritis. Thank you very much, Professor Ahmed Bolad.